Hello, everyone. My name is Bethany Young-Holt, and I'm the Executive Director of the Initiative for MPTs, or the IMPT. On behalf of the IMPT Secretariat staff, I'd like to welcome you all and thank you for participating in today's webinar on opportunities and challenges for long-acting MPTs. We are really pleased to have nearly 100 people registered for this webinar from around the globe. So we have tailored this webinar series for those who might be newer to the MPT field, but we do hope that the discussion is useful for anyone who is interested in MPTs. Overall, we aim through this webinar to outline expert perspectives on opportunities and challenges for employing long-acting delivery methods to develop MPTs that are consistent with the needs of HIV prevention, and defining and developing drug substances consistent with the technical requirements of long-acting MPTs. To start off the webinar, I'll provide a very brief overview of MPTs and the work of the initiative, and I'll then turn it over to our two presenters, Dr. Charles Flexner and Dr. Arnab Chatterjee, both of whom have expertise in the areas of long-acting prevention technologies. During their presentations, we'll be muting the line for all participants. We'll then have time for comments and questions from the audience. Audience members may participate in the discussion through the raised hand feature or type in the chat box, but we will also be unmuting the line at that time so participants can pose your questions directly to the presenters. I'd also like to note that the panelists will be able to stay on for a few minutes after the 60-minute mark, depending on the volume of questions. So I realize that many of you are already familiar with MPTs and the work of the initiative, but I thought it would be helpful for those newer to the field to provide a brief overview of MPTs and the initiative. So MPTs, or multi-purpose prevention technologies, address the interlinked sexual and reproductive health risk and deliver varied combinations of contraception, HIV prevention, and other STI prevention. While a priority for the MPT field has been on contraception and HIV prevention, MPTs do encompass an array of other prevention combinations, namely HIV plus other STIs, contraception plus non-HIV STIs, and combinations of STI prevention, such as HSV and HIV prevention. Beyond the benefits that we see in MPTs for advancing the health of women and girls globally, there are also potential social and economic benefits. There's the potential for greater efficacy in terms of cost and access and delivery of sexual and reproductive health prevention products. So since 2009, the MPT field has grown from a concept to over 24 products in various stages of development, with nearly a dozen MPTs in clinical trials. So while the focus of this webinar today is on long-acting MPTs, I wanted to have this slide to provide a snapshot of the array of MPT candidates and delivery methods that are in the pipeline. These include on-demand and long-acting delivery methods. Some examples of MPT candidates in the pipeline include new vaginal ring designs that are longer lasting and could deliver contraception and HIV and other STI prevention. There are also vaginal gels, films, and tablets that offer different combinations of protection. Injectables and new types of implants are also in development and to deliver contraception and HIV prevention, and these will actually be discussed further during the webinar presentations. So such new technologies and approaches could become part of a suite of products that are accessible and meet the needs of women who use them. So for more information about MPTs that are in the pipeline, you can visit the online MPT pipeline database that is on the initiative's website. So just briefly, the Initiative for MPTs, our IMPT, is a product-neutral international network of experts that are working together to advance this growing field of MPTs. With a long-term strategic vision for the field, the Secretariat facilitates collaboration among MPT stakeholders to optimize product development and introduction efforts. Working to ensure that the product development and work in the field is complementary and aligns with identified research priorities. So in this vein, today we are convening leading experts to help unpack the important topic of opportunities and challenges for long-acting MPTs. So we are just really pleased to welcome our two presenters who will address important aspects relevant to opportunities and challenges for long-acting MPTs. First, we have Dr. Charles Flexner, who will be speaking on employing long-acting delivery methods to develop MPTs that are consistent with the needs of HIV prevention. Dr. Flexner is a professor of medicine in the divisions of clinical pharmacology and infectious diseases and professor of pharmacology and molecular sciences in the Johns Hopkins University School of Medicine. He is also professor of international health in the Johns Hopkins University Bloomberg School of Public Health. Dr. Flexner is an expert on the basic and clinical pharmacology of drugs for HIV, AIDS, and related infections, including viral hepatitis and tuberculosis. His scientific contributions include work on the important roles of pharmacokinetic enhancement, medication adherence, and dosing frequencies in the long-term management of HIV/AIDS. He led clinical development teams for 7 HIV-related new molecular entities, as well as randomized prospective trials of once-daily versus twice-daily combination antiviral regimes. His current research includes the discovery and development of new molecules and formulations for long-acting parenteral administration for treatment and prevention of HIV infection. He directs the long-acting extended-release antiretroviral resource program, also known as LEAP, and is co-principal investigator at the Johns Hopkins University Baltimore, Washington, India Clinical Trials Unit. Dr. Flexner is the Deputy Director of the Institute for Clinical and Translational Research at Johns Hopkins and has served as a consultant for the FDA to the U.S. House of Representatives. So directly following Dr. Flexner's presentation, Dr. Arnab Chatterjee will address defining and developing drug substances consistent with the technical requirements of long-acting MPTs. Dr. Chatterjee is the Vice President of Medicinal Chemistry at the California Institute of Biomedical Research, also known as Calibr, in La Jolla, California. Calibr is an independent, not-for-profit organization focused on translational research with a mission to discover and develop innovative therapies that address unmet medical needs through partnerships with experts in academia and in industry. Dr. Chatterjee's work over the past 15 years has focused on hit-to-lead and lead optimization on several medicinal chemistry projects that address a variety of therapeutic areas, including in the areas of neuroscience, oncology, respiratory disease, and infectious diseases. His research interests and expertise include application of novel synthetic methods to expedite the structural diversification in medicinal chemistry, cell-based lead optimization, and advanced formulation methods for delivery of small molecules. His undergraduate degrees are in chemistry and business from Northwestern and a PhD from Caltech, or the California Institute of Technology. So we are thrilled to have these esteemed experts joining and presenting on this webinar. And before turning it over to Dr. Flexner, I want to remind you that we will have time at the end of the webinar for questions and discussions, and we're really looking forward to hearing from you. During the presentations, if you do have questions, please feel free to send them through the chat function, and we can use them to kick off the discussion. So I will now turn it over to Dr. Flexner.
Thanks, Bethany. I hope everyone can hear me okay. Maybe?
Yes, thank you.
Great. Okay, thank you. So I, first of all, would like to say it's a pleasure to be here with all of you this morning or this afternoon. I'm going to start by talking about some existing long-acting delivery technologies as well as some important technologies in the pipeline, and we'll say a little bit at the end about the potential utility of these products for MPT applications. So I want to start with this question, and I think it's an important one. That is, how close are we to having approved long-acting treatments for HIV? Well, it turns out we have 2 intramuscular long-acting injection formulations for antiretrovirals, long-acting cabotegravir and long-acting rilpivirine, that are already in phase 3 clinical trials. The phase 2B results of 96-week results of studies with these 2 drugs were published last September. And what they showed was that the anti-HIV activity of these 2 long-acting formulations, given together either every 4 weeks or every 8 weeks, were excellent and were comparable to a comparison oral arm consisting of daily oral cabotegravir with abacavir and lamivudine. The technology used for these long-acting injectables is classic nanosuspension of a nanocrystallized drug, similar to the formulation technology used for the long-acting injectable anti-schizophrenic drug paliperidone palmitate. The cabotegravir formulation in particular can produce measurable concentrations of this drug in the plasma for more than a year after a single 800 milligram intramuscular injection, as shown in the red line at the top of this figure. Some potential advantages of injectable long-acting drug delivery systems include the fact that they're well-known, established, and have been used in several different approved drugs that are already available for other applications, including schizophrenia. But they can be inexpensive to formulate because the formulation technology is fairly straightforward. And therefore, this approach is readily applicable to a number of small molecule active pharmaceutical ingredients. In the case of HIV treatment and prevention, these technologies could easily be combined with existing contraceptive injectables, for example, DMPA, one of the most popular injectable forms of hormonal contraception. Potential disadvantages of this approach, however, include the fact that once the injection has been given, it's not easy to reverse the effects of the drug in case of adverse events. In addition, pharmacokinetics in an individual and between individuals can be variable and may be dependent on the injection site. It may not be easy to co-formulate several drugs into a single injection for multi-purpose technologies, particularly if the injection volume is large, as is the case for cabotegravir and rilpivirine, which are currently 2 milliliter injections each at every clinic visit. In addition, the current formulations for cabotegravir and rilpivirine require a 1-month oral lead-in period with daily pills to ensure tolerability before the injections are started. So that's those are the drugs that I think that are likely to be available and approved for treatment of HIV in the United States in the next 12 to 18 months. There's also a large phase 3 prevention or PrEP trial with injectable cabotegravir whose results should be known in a few years. So I want to move on now to novel drug delivery technologies in the pipeline for HIV prevention. And so I'll start by saying a few words about vaginal rings, a topic that Bethany introduced earlier. Vaginal rings have a number of potential advantages, including the fact that they allow for topical drug delivery in theory for as long as several months. They can be easily inserted and removed by the user. There is a vaginal ring already approved for contraception. There is also clinical proof of concept for prevention of HIV with vaginal rings containing the antiretroviral drug dapivirine. And as Bethany mentioned, there is the possibility of pairing an antiretroviral and a hormonal contraceptive in a single device. But potential disadvantages of this delivery technology include the fact that vaginal rings are easily removed. And so for both prevention of pregnancy and prevention of HIV, there needs to be an important education with the user about the necessity of following instructions and not removing the ring at times not dictated by prescribing instructions. And so the effectiveness of vaginal rings is therefore very much adherence dependent, which is unlike injectable technologies or implantable technologies we're going to talk about in a bit. In terms of co-formulation, although this is a possibility, it may be a challenge to achieve the desired drug release properties for both the contraceptive and the antiretroviral, and I think this is an issue that Arnab's going to talk about later. So let's move now to discuss implants for long-acting drug delivery. The advantage of implants include the fact that unlike injectables, they are removable, although it is that would require, in most cases, a minor surgical procedure because these products are implanted subcutaneously. They do provide, however, more consistent and predictable drug release than injectable technologies, and their pharmacokinetics, therefore, are not dependent on the site of injection. Approved implants currently on the market may remain in place for years because they are based on inert, non-degradable polymer materials. And in the case of the levonorgestrel implant for concentration, that product can remain in place for 5 years after a single implant. But there are some disadvantages, including the fact that there is a specialized device required for insertion of these drug delivery devices. There is a minor surgical procedure to remove them. They are regulated by drug development and implementation regulatory authorities as both a drug and a device because the implant itself is considered a device. There could therefore be difficulty in moving these technologies to a generic marketplace because the properties of the device and the release rate of the drug would both have to be matched. In terms of investigational implants for HIV prevention, there is here is an example of a polymer implant developed for the delivery of the antiretroviral drug tenofovir alafenamide. This implant was described in a publication in 2015, and there have been updates on the use of this device in animal models at international conferences, including a presentation last year. This is a silicon-based scaffold with a polymer membrane coating, and the core of this implant is 100% pure drug. And so the drug is released very slowly through pores in the implant after subcutaneous placement. In beagle dogs, these implants deliver tenofovir to the plasma very gradually over a period of several weeks. And so what you can see in this figure, in the middle of the figure are the plasma concentrations of tenofovir, the nucleotide reverse transcriptase inhibitor, Below that are concentrations of the prodrug, tenofovir alafenamide, and at the top are intracellular concentrations in peripheral blood mononuclear cells of the active metabolite, which is tenofovir diphosphate. And you can see in the case of all three of these entities, a very slow release over a period of 40 days, and the possibility that one might be able to deliver effective anti-HIV concentrations of TAF from such an implant for several months. Another investigational antiretroviral nucleoside that has been tested in implant form is EFDA, an adenosine analog, also known as MK8591, being developed by Merck. And here is a figure from a preclinical study carried out with this drug delivery system that was presented at an international conference in 2016. And what this figure shows are the concentrations of the active drug, EFDA or MK8591, in the plasma of rats after a single implant over a period of, in this case, 180 days. And what you can see here is that using two different formulations, designated here as formulation 1 or formulation 2, EFDA could be delivered into the plasma of these rats at a nearly constant rate with very slow decline in concentrations over time in this experiment for a period of 6 months. And this drug delivery device looks like it could have the potential to deliver antiretroviral concentrations of EFDA for perhaps as long as a year or more. This is just a reminder that there is proof of concept for this kind of implantable delivery system used to prevent, in this case, pregnancy over a period of several years. And in this figure, now we're looking at the plasma concentrations of two different hormonal contraceptives, etonogestrel and levonorgestrel, delivered into the plasma after a single implant in women, with the levonorgestrel concentrations shown in the closed bars at the bottom of the figure and the etonogestrel concentrations in the open bars at the top of the figure, showing more or less constant delivery of both of these contraceptive hormones to the plasma for a period of several years. And as I mentioned earlier, in the case of levonorgestrel, a constant delivery of this hormonal contraceptive to the plasma for as long as 5 years after a single implant. And now we're looking at technologies for HIV prevention and hormonal contraception that could, in theory, be combined into a single implant. I want to talk briefly about broadly neutralizing monoclonal antibodies, because they are a new antiretroviral biological approach that does in fact have promise for long-acting delivery and could be used for HIV prevention. So this is just a figure from a review published in 2016 in the New England Journal of Medicine, reminding us that there are many different possible binding sites on the HIV envelope protein, GP160, that would result in a broad neutralization of HIV isolates. And a number of these monoclonal antibodies are shown in this figure, but there have now been several dozen of these isolated, and approximately 7 or 8 of these taken into clinical trials. One of the nice things about monoclonal antibodies is they can be converted to a long-acting form with specific mutations in the Fc receptor portion of the antibody. And that, in fact, has been done in this case with the monoclonal antibody VRC01LS, LS standing, designating the long-acting version. And this is a publication from PLOS Medicine from January showing the pharmacokinetics of VRC01LS in healthy human volunteers after a single intravenous infusion or subcutaneous injection of this antibody at 3 different doses. And in all cases, what you can see is that there are antiretroviral concentrations of this monoclonal antibody in the plasma for at least 6 months after injection into these healthy human volunteers. So here's another potential tool for combination technology. Advantages of broadly neutralizing monoclonal antibodies include the fact that in this case, not only are they humanized, they're actually derived from antibodies taken from human subjects. And so they are very well tolerated and are not very immunogenic. As I mentioned, their pharmacokinetic profile can be extended by a simple molecular modification. And they can also induce beneficial host cell-mediated immunity, including antibody-dependent cellular cytotoxicity responses. There is currently a large 2,500 patient study of the immediate unmodified version of VRC01 taking place in South Africa. And that study is almost fully accrued using that antibody as a sole agent for HIV prevention or PrEP. Potential disadvantages of these molecules, however, include the fact that at least at present, they're relatively expensive to manufacture in large quantities. They require an intravenous route of administration in most applications, although subcutaneous administration is also possible. We know that there's pre-existing resistance in many individuals, even those who have never been exposed to these antibodies. And so when these antibodies are used as monotherapy in infected individuals, they do and they will select for resistant viruses. And that's a problem that will require either combining antibodies or combining antibodies with small molecules in order to prevent this issue. What about oral dosing of long-acting technologies? I want to finish describing a recently published oral reservoir device for delivery of small molecules, and this, as shown on this figure, is essentially plastic star-shaped device with an elastomeric core and rigid plastic polymer arms, each of which can be embedded with a different drug. And so one of the nice things about this kind of a device is it's perfect for MPT applications. The device itself is swallowed and is too large to be passed out of the stomach and can remain in the stomach for 7 to 14 days in its current iteration, releasing drug throughout that period of time. Eventually, gastric acid breaks down the connector pieces of the arms to the core, and the device then falls apart and exits through the intestinal tract. This is a comparison of the pharmacokinetics in the plasma of pigs of 3 different antiretroviral drugs, dolutegravir, rilpivirine, and cabotegravir. At the top are the plasma concentration profiles of these drugs when given in their immediate release oral form, and at the bottom are the concentrations when delivered from this gastric reservoir. What you can see is obviously sustained release over 7 days of all 3 drugs from this device, although the fact that drug concentrations begin to go up at the end of the 7-day interval and also display some variability suggests that there are still some potential engineering problems that would need to be solved in order to get more predictable drug release. So some potential advantages of such a drug delivery device include the fact that it would be convenient and self-administered because it's swallowed by the user. Multiple antiretrovirals or antiretrovirals and contraceptives could be embedded in a single device. There's spontaneous degradation of the device for gastrointestinal elimination. If there were adverse events, the device could actually be removed by endoscopy. So it is removable, unlike some of the injectable technologies we discussed earlier. But potential disadvantages include the fact that the published device I just showed you is quite large for oral administration and has a dosing interval limitation of 1 to 2 weeks. And I think for the kind of long-acting applications we're now interested in, we'd like to see that dosing interval be extended. The gastrointestinal tolerability in humans is unknown because the device has not yet been tested in humans. And there is, of course, the possibility of gastrointestinal ulceration or even obstruction, as well as unknown food and antacid effects. So that's all I wanted to say to you this morning. I do want to leave you with this resource. I am fortunate enough to be directing a National Institutes of Health funded research resource, the Long-Acting Extended-Release Antiretroviral Resource Program. And this is the homepage for our website, www.longactinghiv.org. And for those of you interested, this is a wonderful compendium of resources to learn more about the current state of development of long-acting antiretrovirals for HIV treatment and prevention. And I will close there and turn the floor over to Arnab.
Dr. Flexner, thank you so much for such an extremely rich presentation. I think personally I'll never look at starfish the same. Now we would love to welcome Dr. Arnab Chatterjee, who will be addressing defining and developing drug substances consistent with the technical requirements of long-acting MPTs. Thank you.
Great. I hope everyone can hear me okay. Good morning, good afternoon, good evening, depending on where everyone is joining the call today. It's my pleasure. Really wanted to thank Bethany and the rest of the group at IMPT for putting this together. I think it's been excellent for me to be involved and also to get a chance to talk on some of our work and some of the work that's going on in the field, along with Charlie, who's obviously done a tremendous amount of work in this field. So it's a real honor to talk to everyone today about some of the chemical considerations, I would say some of the medicinal chemistry considerations about how to pick molecules, actual chemical structures of things that may be best suited for this type of approach, not only in the context of HIV, but also in the context of reproductive health, to be able to potentially down the road, as Charlie outlined, kind of try to combine agents in a way that both the pharmacokinetics and the benefit to people receiving this is something that we can envision as a drug product that can then move forward. So I'm going to be using the laser pointer. I hope everyone can see it. I'm going to go through some details on this, but before I jump into details, I wanted to give you a key outline of kind of what I'd like to get across in the next few minutes in my presentation, and hopefully can stimulate some discussion afterwards on kind of what are some of the thoughts of folks more broadly in this area. Initially, I just wanted to introduce Calibr and also our close association and with Scripps Research Institute, which may be more familiar to folks in the audience. That'll be relatively quick. And then I really wanted the key elements of this presentation today are in how do we select the appropriate compounds. Charlie outlined several very nice compounds between carbotegravir, rilpivirine, TAF, et cetera, in the HIV space and obviously in the contraception space as well. how do we pick the right sort of molecules? What sort of properties are we looking for in drugs to be able to really use them and move them forward as good candidates for multipurpose prevention technologies? And much of that has to do with what I kind of have envisioned as being a very delicate balance between formulation science and synthetic organic and medicinal chemistry, where unlike a traditional drug discovery, small molecule drug discovery program where we would generate hundreds, and in some cases thousands of molecules as potential NCEs and test those via different testing funnels to be able to get to candidate molecules to move forward with. In this case, we're actually making a relatively small number of compounds, but really coupling that information and those chemicals to formulation principles, and specifically thinking about how can we modify drugs to make appropriate prodrugs, and to really front load that. So I think the number of new molecules we make is not hundreds or thousands like we would in a traditional oral small molecule program. Here we're actually making 20, 30, 40 compounds, but we're really coupling that to a variety of different formulation technologies and formulation principles, some of them outlined earlier by Charlie, around how can we best deliver those molecules, how can we best formulate those molecules in the context of pharmacokinetics, duration of action, compatibility, cost of goods, etc. The second point I wanted to highlight here is how important it is to understand the mechanism of action and how important that mechanism of action, not just in terms of resistance profile, but also in terms of how much drug do we need to get to target tissues, target cells, and what is intrinsically the potency and clearance out of those compartments beyond just thinking about plasma. And then finally, in the last part of the presentation, I wanted to highlight what some of the key research gaps and priorities are. My lab is actively working in this space in collaboration with the Gates Foundation and with Medicines for Malaria Ventures in the areas of HIV and malaria prevention. And how do we try to address some of these issues that have come up in terms of potency, clearance, volume of injections, etc. And also, finally, as a little bit of a forward-looking statement, talk about how one may actually want to consider new chemical matter as being the right sort of compounds to move forward in these applications versus just drug repurposing. So I think to date the field has been very, I wouldn't say limited, but has been very opportunistic in taking the existing compounds that have known safety profiles in humans, in many cases known efficacy profiles in humans, and really sticking to those molecules versus thinking about how does one actually come up with chemical entities that are actually tailored for this approach, where the pharmacokinetics profiles do not require an oral run-in, or where the formulation principles and clearance properties of the molecules are completely independent of oral bioavailability, which is typically how these molecules have been optimized to date. So with that overview, I just wanted to jump in a little bit and tell you a little bit about Calibr. I am on slide number 40 on my end, but it doesn't appear to be refreshing on my end. I hope everyone can see slide 40. Ah, great, okay. Sorry, a little bit of a hiccup there. On slide number 40, I just wanted to outline a little bit about Calibr. So this institute is about 6 years old now, and started by Peter Schultz, who is a chemical biologist, physical organic chemist by training. He established an institute that is now part of Novartis's research organization. We are a relatively small institute of about 100 to 120 scientists. We have collaborations with a wide variety of groups, originally funded by Merck, and then subsequent funding from the Gates Foundation and other for-profits as well as nonprofits. And as shown on the lower right-hand side, we've now, as of 2016, are a subsidiary of Scripps Research Institute, which is one of the largest nonprofit biomedical research institutes in the U.S., and primarily the goal of Calibr in its combination with Scripps is really to be able to translate basic research into drug discovery and into clinical opportunities. And we are very fortunate to have 3 molecules now that have moved into clinical development in the last few years, and we anticipate another 6 to 8 programs to do that in the coming 12 to 18 months. Again, the goal of Calibr is really to bring together not just small molecule science, but also biologics, engineered cells, et cetera, to be able to meet a gap in requirements for patients for unmet medical needs. So on slide number 41, I just wanted to highlight the fact that what we've been able to, or what we envision is the eventual goal of the combination of Calibr and Scripps is really to provide a new model for bench-to-bedside nonprofit research. And again, much of that is centered out of a long-standing history within Scripps Research Institute of doing drug discovery, ozanimod, Humira as 2 examples, but really to couple that to the engine of doing preclinical and early clinical development of organizations to move forward multiple assets into the clinic. So with that brief overview about Calibr and Scripps, I wanted to jump right into a little bit about the intramuscular approach. And so on this slide, really the key element of what I wanted to talk about, and a little bit of this is highlighted in some of the work that Charlie presented earlier, is how can we be able to take a typical oral drug, which has a profile of a relatively high Cmax, relatively quick onset of plasma concentrations and drug distribution to relevant tissues and cells, and a relatively fast clearance in that compartment versus kind of what we would like to be able to do, which is more of the brown and certainly the purple line, of being able to get to beyond a typical oral drug to get to a more atypical drug, which would allow us to have a long period of coverage. And as Charlie outlined, an oral run-in is typically required for molecules to be able to get that kind of duration of action over a long period of time. So on the right-hand side here, I just wanted to show what kind of the ideal PK profile is, which is a relatively quick onset, 3 to 4 months of coverage in some cases, or even longer, though I would say that the field is now thinking a little bit more detailed in terms of duration of action of an intramuscular depot, what kind of length of time is appropriate, and how long is maybe too long for what a patient would eventually want versus something that is just too short and reduces the compliance, which is obviously the issue with taking oral medications on a daily basis. So in being able to generate that window between the effective concentration, or what's called the MCC, or the C effective concentration, and the toxic level. So how can we flatten the PK curve in a way such that we are well above those toxic levels, given that we are, in many cases, using these as preventative technologies, which require even a greater safety bar. So with that in mind, from a pharmacokinetic standpoint, what we have envisioned here is actually in many ways been precedented over many years in a variety of different disease areas, is thinking about what sort of partitioning of small molecules would need to happen between an oil and water interface. And the real interest in doing that is being able to understand how there are 2 general approaches to be able to get to molecules that can be then applied for long-acting depot technologies. So the first one is a relatively new field, and I think it was highlighted a little bit by Charlie, where an aqueous suspension approach is used, either a nanomilled material such as carbotegravir or rilpivirine, or a micronized material, which has been more precedented, and be able to have a suspension formulation that allows for high drug loading concentrations. That drug would release from the depot at a very small rate. There may be secondary depot sites that are generated, and at that point, small molecules would be able to go into circulation, and given an intrinsic clearance, what we're calling here is the CL, then leads to elimination of the drug. And it really is that dissolution of the small molecule out of the injection site, the dissolution of the nanoparticle or microparticle that then can release drug into into systemic circulation. The other approach, which is a very historically used approach, injectable testosterone is one very well precedented example here, where one can actually use a small molecule that forms a very oil-soluble lipophilic solution. And in a lipophilic solution, typically we're taking advantage of small molecules that have very lipophilic characters, so compounds that are very poorly water-soluble but highly soluble in oils and fats, and be able to either attach a prodrug or some sort of chemical functionality to that drug to be able to allow slow release out of the depot. So where an oil would be sitting in an aqueous environment and then being able to slowly release the drug. And there are unique challenges to both of these approaches, very much centered around the molecules and how the loading of the drugs, meaning how much drug is there per unit volume, what the melting point of those molecules are, such that the melting point can serve as a potential predictor of dissolution rate out of a particular particle or out of a particular environment that the particle is sitting in, be it an aqueous environment or an oil-based particle, and really can affect dissolution out of that based on particle size. And some of that has been demonstrated with carbotegravir and ropinirole, but other aqueous suspensions, including antipsychotics, where the particle size is dramatically related to the rate of release out of the depot and allowing you to have a long-term coverage of small molecule over time. So with that, I'll jump into a little bit of the chemistry of kind of what typically can be done to molecules to be able to allow them to be even better candidates for a multipurpose prevention technology, particularly as an IM depot, but one can also imagine this technology could be used for other types of devices, implants, rings, etc. But typically we're using prodrugs, and historically prodrugs have been used many times and often in medicinal chemistry. As a matter of fact, a large number of the drugs that are in clinical trials and available to patients are typically functionalized as prodrugs. And one very widely used example of prodrugs is certainly in the area of beta-lactams as antibacterials, where prodrugs are used to improve permeability across many different types of biological compartments, that being the gut lumen for molecules that are poorly absorbed, getting molecules across the blood-brain barrier, molecules to target cancer cells, etc. And those sort of prodrugs have been used to really improve bioavailability to all three areas here, both across the gut, into the CNS, and into target cells using prodrugs. In HIV, for example, prodrugs have been used extensively. I think the most recent generation of prodrugs is really kind of chemical functionality called phosphoramidites, which I'll get into the next slide, that's used for molecules such as tenofovir alafenamide, which not only improve target cell levels, but also improve clearance also improve overall clearance of the molecule by reducing the clearance from the target cells. In this case, it's PBMCs and CD4 positive cells. And how can one actually administer drugs that are not just simple prodrugs that improve bioavailability, but how can we actually use prodrugs to be able to improve bioavailability to the target cells? And phosphoramidites and the use of the ProTide technology that's been developed in the UK over many years has really been one area that has had a dramatic impact in the area of HIV. And again, one thing I wanted to highlight in this presentation is that taking oral drugs and using them as in a parenteral formulation is not necessarily very easy. And it's similar to work in biologics, vaccines, and et cetera, where in many cases we need to be able to optimize the molecule, specifically the chemical entity, to be the best approach for a parenteral approach versus a simple, versus what has been developed for oral therapies. So with that, I'll jump a little bit into the chemistry of tenofovir alafenamide, which I think is a very interesting molecule. As shown on this slide, I show kind of the first generation tenofovir-based prodrugs. You'll notice that the tenofovir piece, which is on the right-hand side of the molecule, is conserved between tenofovir disoproxil and tenofovir alafenamide. But tenofovir alafenamide is different in the nature of the left-hand side of the molecule, where a phosphoramidite is used as a prodrug approach versus a biscarbonate-based phosphate ester, which is used in TDF. And what we now know is that the therapeutic index, as well as the overall clearance and potency profiles of phosphoramidite-based prodrugs, is superior to that of tenofovir disoproxil in the context of both hydrolysis to tenofovir monophosphate, which is shown here on the right-hand side, which is known to be a toxic metabolite that can be present in plasma and affect multiple tissues. It limits the ability of formation of tenofovir, TFV, in plasma, and actually dramatically increase the concentration of tenofovir diphosphate, which is shown in the red box, which is the active diphosphate metabolite, which is a direct inhibitor of the biological polymerase target inside target cells. And so this just shows that there's a believed approach through enzymes that are present either within PBMCs, for example, various cathepsins, as well as particular enzymes that may be in the liver that can cleave this phosphoramidite prodrug to then generate a intermediate here that can then undergo cyclization under acidic conditions, both in lysosomes in target cells, and be able to form a cyclic phosphoramidite that then inside the cell generates TFV versus just simple cleavage by plasmasterases to form TFV. Then that TFV inside the cell is then diphosphorylated to form tenofovir diphosphate. Again, an approach which allows for greater concentrations of the active metabolite in the target cells. And again, I think this is of clear relevance not only for oral technologies, which is obviously where TAF is currently used in patients, but also as an approach for being able to make improved prodrugs that can have greater application for MPTs. So with that, I just wanted to jump to, in the next slide, a case study of some work that's ongoing in my lab, which is unpublished to date, around prodrugs for malaria chemoprophylaxis. And the reason why I bring this up is because I think many of the medicinal chemistry principles that we've applied in the context of atovaquone can certainly be applied in the area of HIV, though I will say that there are some significant challenges in the way that anti-malarials have been developed, typically with molecules with very long half-lives in humans versus anti-HIV molecules or HIV preventative molecules typically have a shorter half-life in humans. But with that caveat taken into account initially, we can show that we can actually take a molecule which is atovaquone, which is an orally used malaria chemoprophylaxis drug. It's used in combination with proguanil, as Malarone is the most commonly used chemopreventative measure orally for folks who are in malaria visiting or in malaria pandemic areas. In this case, I simply wanted to show, and these are two graphs that just show the pharmacokinetic profiles of three different molecules. The red line is actually what happens if you take atovaquone, which is a very high melting, very lipophilic drug, and you reduce the particle size to micronized, or in some cases submicronized material, the nanosuspensions of material. And you can actually see in the red line, and this is again PK data preclinically in the rat, where you actually see, and I just show a insert on the right-hand side showing a linear scale, so you get a sense of what the initial drug release out of the depot looks like. And try to prevent things such as dose dumping, for example, where you essentially just see a large amount of drug released within the very short period, which then gives you more of an oral pharmacokinetic profile versus a parenteral or pharmacokinetic profile. And you can see that the red line, which is atovaquone itself, actually is cleared relatively quickly. It actually goes below the target concentrations that we think is required for malaria chemoprevention in man within about 3 weeks. And then the subsequent prodrugs that we've shown, which is a blue line here, which is a lipophilic prodrug of atovaquone, and the green line, which is also a prodrug of atovaquone, but in two different formulations. So the blue line is a lipophilic prodrug, which is highly oil-soluble and is completely miscible in oil, versus the green prodrug, which is not miscible in oil, but does form a high-concentration aqueous suspension. And you can actually see by just the nature of the prodrugs that are used, you can see a very very different pharmacokinetic profile, where now the flattening of the Cmax happens very quickly, and you can actually see that the Cmax you achieve in the depot formulations of the prodrugs is about threefold or greater lower than that of atovaquone itself in a similar suspension. And you can actually see that now, because you're not dose dumping and releasing a lot of drug within a short period of time, you're actually generating a prodrug now that has a very long release of atovaquone out to at least 4 months in the case of the lipophilic solution. Finally, I want to mention that while it's important to show that prodrugs can have a dramatic effect in terms of pharmacokinetic profiles to be able to lead to a target product profile that is most consistent with whatever molecule you may be combining this with down the road, that the drug stability in that formulated material is very important. And I think one of the key research gaps to close is really the need for understanding drug stability, prodrug stability, drug stability, both in terms of chemical stability, formulation stability, to us to be able to eventually combine these with other agents that can then lead to very effective multipurpose preventative products. Finally, with just a few minutes that I have left, I'd like to point a few things for future considerations. And one thing that I alluded to earlier is the need currently of being able to stick to oral drugs, or should we actually place great importance on novel candidates? I mentioned that only because as a medicinal chemist throughout my period of time working on active lead optimization programs, we typically leave a lot of very good compounds on the shelf that may actually be really good IM depot candidates. And while we're trying to leverage a lot of the clinical safety and efficacy of molecules in the MPT field, we typically are generating a lot of molecules that may actually be better candidates for IM depots based on compounds that have poor aqueous solubility, and in many cases may actually have better clearance properties and potency properties that may be amenable for MPTs. And so that's one thing I wanted to make very clear in this presentation, is that this is one area where I think the field is moving to, given that not a lot of molecules meet the kind of potency, clearance properties as a parenteral drug, contrary to the way that they were developed initially as oral medications. The other thing I wanted to point out is that it's exceptionally important to think about chemical modification. Prodrugs have been at the very heart of the way that many drugs have been developed for oral delivery and to be able to get molecules to target cells and across biological membranes. And that's a really important approach to bring to bear for the MPT approach, given that there's a great expansion in formulation technologies which make certain types of drugs, certain levels of molecular weight, lipophilicity, particle size, melting point, etc., which then allow us to build a really customized solution for the MPT approach. And again, as I mentioned at the very beginning of my presentation, combining drugs and drug formulation is really the way that the optimization process has been done, certainly not only within the Calibr, but also within other areas of folks who are working in this space. And really coupling formulation science to synthetic chemistry is really at the core of being able to bring the best technologies forward to these various populations of folks we want to be able to help. And with that, I want to thank again for everyone for taking the time today to allow us to present some of our work and thoughts in this area.
Arnab, thank you so much for your great presentation, and both of you. We will now move on to the question and answer portion of the webinar. I am seeing that we are almost to the hour. So if you do need to leave the webinar before the discussion and question and answer time, we will have the recording. We will have a recording of the webinar and the slides, so we can circulate that and make that available. But we do hope people are able to stay on. For those who do have questions or comments, we'll ask that you please use the raise hand feature, state your name and your affiliation and your question. You can provide a question or comment in the chat box on your webinar screen. So as the audience gathers your questions and thoughts, I actually have a few questions of my own. So I'd like to get the discussion started with a question, and then after the participants answer this first question, we will unmute the line. I will just say that once we unmute, if you don't have a question, just to cut down on background noise, if you can mute yourself. So So in thinking about co-formulated long-acting MPTs in the primary target populations in which MPTs may have the greatest impact, can you, Arnab and Charlie, speak to the issues that we need to consider around the compatibility and duration of action of preformed drugs that would be potentially combined or co-dosed in the context of an MPT? Maybe Arnab, we can start with you.
No. I mean, I think one of the things that we have paid a lot of mind to, and I think in the malaria space is one such example, but also in HIV as well, is that typically what we have been able to do is front load many of the drug stability studies, which I kind of outlined at the end of the presentation, where we're really considering what is under kind of accelerated conditions, for example, especially in parts of the world where we want to be able to reach with these types of approaches. What is the shelf life stability of molecules, give it prodrugs, give it particles, etc., and what is the stability in various types of excipients. We can see very subtle differences through the additions of additives, for example, that can reduce the increase in particle size over time, or the effect known as Ostwald ripening, versus the intrinsic stability of prodrugs in various excipients, and really be able to couple that with molecules in parallel. So, for example, in the malaria space, one certainly would given the resistance frequency of the Plasmodium parasite, we would certainly want to move forward with the combination, and we certainly would want to be able to co-formulate two molecules together. So we are very much focused on the ability to take a limited set of excipients, in this case just as an IM depot approach, take a limited number of excipients, find compatibility of molecules together in that formulation to make sure that there's no effects of those two molecules being in the same injection, and then moving forward and understanding the stability of both of those molecules independently, with the eventual goal that those will be combined again as the eventual drug product.
Great. Charlie, do you have anything to add to that?
No, I agree with that really excellent summary of, I think, what is one of the most important challenges in creating MPTs, and that is the compatibility of the active pharmaceutical ingredients, and also choosing agents that have matched pharmacokinetic properties. So, for example, if you were trying to develop a single device for HIV prevention and prevention of pregnancy, it wouldn't do you much good if the contraceptive component of the device would work for 5 years, but the HIV component would only work for 6 months. And so I think if we're going to create delivery devices that really fill the niche we believe is out there, I think we're going to have to work on making sure that the antiretroviral component and the contraceptive component have similar release rates and pharmacokinetic properties, as well as some of the chemical instability issues that Arnab talked about.
Yeah, great. Thank you. So we are now going to open the line for participants to ask questions or share your comments. If you are not speaking, please mute your lines, but if you've connected over the phone with Ready Talk, you can press *6 to mute and press *7 to unmute. If you have dialed into the audio through your computer, you can also mute yourself using the mute button on the webinar screen. So does anyone have a question? I don't see any raised hands, but I'll...
The conference has been unmuted.
Great. Does anyone have a question? Okay, if people are still getting your questions together, I have another question. So given your experiences, Charlie and Arnab, do you see opportunities to harness current research on long-acting ARVs for HIV prevention to help advance the MPT field? For example, do you have thoughts on how lessons learned from long-acting delivery technologies being evaluated for HIV prevention currently can inform and enhance the prospect for next-generation long-acting MPTs? So, Dr. Flexner, we can perhaps start with you, but I would like to, I'm hearing some background noise. So as a gentle reminder, we would like to leave the line open for participants, but please mute your line. You can press *6 to mute, and if you're not speaking. But if this is really unsurmountable, we'll have to mute the line. So I'm still hearing, so if you could all check your lines. So, Dr. Flexner, would you like to respond? Did you hear my question?
Yes, I did. And let me just say that I'm still hearing a lot of feedback. I don't know if you want to mute the lines. Hello?
Charlie?
Yeah, did
yes,
you mute?
You can go ahead.
Great, okay.
Yeah.
Good. So I will say there has been a lot of back and forth in terms of learning and advancing this field from looking at agents that are already approved and in use for other purposes. So, for example, I think the approaches to long-acting rilpivirine and long-acting cabotegravir were based, at least in part, on the approaches developed for long-acting injectable anti-schizophrenic drugs. Particularly paliperidone palmitate, which uses, as I said earlier, very similar chemistry and similar delivery strategy. In addition, I think we have a lot to learn from the hormonal contraceptive field, both in terms of injectables and implants. I think, on the other hand, those fields are likely to learn a lot from what we're doing in terms of the possibility of combining multiple agents into a single delivery strategy. So I think there will be a lot of impact of what we're doing in the antiretroviral field on hormonal contraception and the prevention and control of other important infectious and non-infectious diseases. And Arnab's example of malaria, I think, is just one of several. We also have quite a very active area on the development of long-acting drugs for prevention of and eventually treatment of tuberculosis. And there is also discussion of applying some of these same technologies to prevention or treatment of acquisition of hepatitis C virus.
Thank you. Arnab, do you have anything to add to that?
Yeah. I think certainly Charlie's summary is on point, and I think there are certainly applications from these areas that have a more historical field in the approach. I certainly think that some of the things that have been used, for example, in the contraceptive fields, in the testosterone example that I mentioned before, of molecules that are exquisitely potent. The potency of these molecules is in many ways unprecedented, and how to be able to really be able to think about potency in a very thoughtful way to be able to get to the right sort of candidates for an MPT approach. And certainly more recent technologies that are present, that are even present, for example, in the areas of slow release of drugs, of even peptides and biologics, for example, through using various types of approaches, is certainly something that can be applied to the MPT field as well. I think obviously cost of goods is a serious consideration there, but I think those technologies, as they kind of move forward, may actually be really good candidates for being able to take forward some of the sort of molecules that we think are some of the best classes of molecules or mechanisms of molecules that we would like to use for the MPT approach.
Great, thanks so much. Does anyone in the audience, we did have to mute because of the background noise, but if anyone has a question, please enter it in your chat box. I do. You mentioned just now, Arnab, you brought up cost of goods, and I'd like to sort of pick up on that with another question that I have. Given limited resources for product R&D, how would you recommend addressing cost of goods and the selection of compounds and delivery methods, and how new technologies can help push down costs? This is clearly going to be an important consideration given just finite resources. So Arnab, do you want to start with that?
Sure. I mean, I think one of the things we always kind of talk about, and historically, a lot of the work that's been done, at least in the neglected infectious disease field that I've had the opportunity to work in for several years now, that has certainly been a major consideration, and something that we have in many ways tried to address preclinically and maybe even in the early stages of programs. But I will, for example, the example that Charlie showed of monoclonal of an antibody approach versus a small molecule approach, there are some distinct advantages that one could take advantage of. Certainly, in terms of delivery devices and delivery formulations, I think there is a great amount of work that's being done in bringing down the cost of, for example, even aqueous suspensions, given that in many cases the requirements for cold chain, the requirements for a variety of stabilizers to be able to keep an aqueous suspension relatively inexpensive compared to an oil-based solution, which in many cases is many-fold less expensive just by the nature of the fact that the molecule is in solution, is something that clearly there is a gap right now, and there are ways to close that gap in terms of new work that people are doing, for example, in stabilizing suspensions. So I think that's certainly a major area of consideration. I think it's certainly something that, as we come up with better compounds and better formulation technologies, that those two things together can reduce the overall cost. But given that the impact here, both for in the area of contraception, but also in the antibiotic area, be it for HIV, TB, malaria, et cetera, is significant, such that I would not. My general impression is that using that as a very early consideration is probably not the best use, rather than trying to find the best molecule and the best formulation and the best device. And then as we learn more and more about new emerging technologies to be able to reduce cost of goods, is something that we should be able to involve earlier in the process, but not to the point where we don't undertake interesting projects.
Thanks so much. Charlie, do you have anything to add to that?
I'll just add that I think there's a general misperception that long-acting technologies are a more expensive way to deliver drug per unit of time, and there are a number of examples that clearly correct that misperception. So, for example, if you look globally at the most cost-effective form of hormonal contraception, right now it's levonorgestrel implant, which costs around $15 for a single implant and provides effective contraception for up to 5 years. So the cost per day for effective contraception with that kind of a strategy is amazingly inexpensive. It's pennies a day. So I think there are good examples out there of how long-acting technologies can, in a number of cases, actually make drug delivery less expensive, not more expensive.
Great, thanks. That's great. So here's a question that was posed by Joe Romano. The question is that the question of what is long-acting has largely been addressed in contraception, as evidenced by the established duration of effect for various contraceptive products. For example, there are contraceptive implants that are effective for 3 or more years. There are 1 milliliter injections that last 2 to 3 months, IUDs that are 5 or more years have effectiveness, and then there's contraceptive rings are being developed. So presently, the rate-limiting issue for long-acting MPTs is HIV prevention. Is the HIV prevention component. So could you speak to how far the field is from having an MPT that is contraceptive and prevents HIV infection for a duration that's consistent with the presumed target population's experience with contraceptive-only products? And then also, what are the key technology issues that need to be addressed in order to expedite the timelines towards achieving such MPT products? So would one of you like to start with that? Maybe Charlie?
So, I think there are certainly vaginal rings in development and testing right now that have both an antiretroviral and a hormonal contraceptive. And as I said before, the target interval for those rings is up to, it's one ring per menstrual cycle, or and rings can be used for up to 3 months. So I think that's consistent with a definition of long-acting, and those are devices in development that appear to have the possibility of delivering both effective HIV prevention and pregnancy prevention. When you talk about what's the definition of long-acting, I do think the field is evolving. And I think most of us would say that technologies we're looking at right now need to have a dosing interval of 1 month or longer in order to be truly long-acting. And so that may seem like a low bar for pregnancy prevention, but it's a game changer for HIV prevention. And so, I think we're getting there. We certainly have LA cabotegravir and VRC01, which are both being given every 8 weeks for HIV prevention, and would certainly either be combined with contraceptive technology.
Great, thanks so much. Arnab, do you have anything to add?
Yeah, just one point there. I think it's a really important point that Charlie brought up, is that being able to match the PK profiles is not just in terms of longer and longer and longer, but rather being able to titrate the pharmacokinetic profile to best match the eventual use of the drug, be it giving the duration of action that people would be looking for. Some may want shorter durations, some may want very long durations. And so I think the ability to take a particular molecule or particular mechanism of action and be able to dial in that kind of 1 month, 3 months, 6 months, 1 year, greater than 1 year is something that is attainable for at least some of the molecules that have been reported to date, and that needs to be put into close consideration, not just for the contraception piece, but also for the antiretroviral or anti-infective component.
Great, that's very thoughtful. Thank you. So we have a question that came into the chat box from Marjolein Van Vliet, she's a graduate student from the Netherlands. And she thanks you for your presentations. She's wondering about your thoughts around the potential uptake for injectable long-acting forms of PrEP by women using injectable contraceptives like DMPA. Specifically, what are your thoughts about the proportion of women who would be willing to start with extra injections? So I think what she's really getting at is like co-administration of contraceptives like DMPA and PrEP. So she'd like you to think about things like low perceived HIV risk among women. So, yeah, thank you so much, Marjolein, for this question.
So this is Charlie. I can take a stab at that. Certainly, with the clinical trials in which HIV-infected individuals switch from daily oral therapy to injectable therapy, they do require 2 injections at every visit, one of rilpivirine and one of cabotegravir. So those drugs have not been co-formulated. And so the idea that you could come to clinic and get an additional injection of an antiretroviral at the time you got your DMPA injection, I think it's a reasonable strategy. I can't predict what the expected uptake would be because I'm not aware of anyone, doing a needs assessment for that. But I think it's certainly feasible. And the idea that somebody would come into the clinic and get 2 intramuscular injections instead of 1, we have proof of concept for that with the rilpivirine and cabotegravir phase 2 and phase 3 studies, and those studies combined involve more than 1,000 HIV-infected individuals getting drug for more than a year. So I think it's feasible. Whether it's something that is practical and that people would actually embrace, I think would require some work in order to answer intelligently.
Yeah, this is Arnab here. If I may add to that. So thanks a lot for the question. I think certainly additional injections and whatnot have unique have additional challenges for what patients would actually want. My only comment to that is that similar to what I mentioned previously is that I think the ability to get the sort of pharmacokinetic profiles that people are interested in, and I think for contraception certainly long is good, but the ability to be able to titrate that based on needs of particular individuals, I think is a very important consideration to keep in mind. I think that as the HIV field, for example, with cabotegravir and rilpivirine is really kind of laying the groundwork for what future clinical trials in this space will look like, it is important to note that as we are expanding the duration of our antiretrovirals, that does need to be matched not only in the context of contraceptives, but also in the context of how long someone would want to be able to have those drugs on board. And I think certainly in the area of antipsychotics, which is a much more precedented space, that there is a sweet spot for many patients in terms of their interest in being able to have a 1 month versus 2 month versus longer coverage of those molecules, both in terms of safety as well as in terms of efficacy, and the ability to be able to be able to get those in the appropriate patients at a reasonable time. So I think that is clearly an area of additional work that needs to be done. And as Charlie mentioned, I think some additional work around kind of what are the expected use cases that would be able to get people interested in being able to get an additional shot or an additional increase in volume rather than it being separate shots. A 1 ml injection versus a 250 microliter injection versus a 4 ml injection certainly has different challenges in terms of pain and tolerance for the patients.
Everyone. I really want to thank both Dr. Flexner and Dr. Arnab for your excellent presentations and really for your willingness to stay on an extra half hour for the question and answer. This has been just a fabulous webinar, and we have it recorded, and the presentations will be circulated as well. So I don't see any additional questions that have come in. So with that, I would like to wish everyone a wonderful rest of your day and evenings. Thank you so much.
Thanks, everybody.
Bye-bye.
Bye-bye.
Yeah, thanks, everyone. Wonderful talking, and thanks again.
Thank you. Bye-bye.